Okay, so that's just an overview of the cells that are associated with alveolar bone and the counterparts that are associated with cemento. I want to have you just make a note that remember we talked about in the PDL, there are also those stem cells, those cells that have not specialized yet, and they can be triggered to specialize under certain circumstances. So they could be triggered to form osteoblasts, and they could be triggered to form cementoblasts to help regenerate tissues. Okay, so when we look more closely at the cells associated with bone, we'll be able to recognize them pretty easily. So the osteoblasts, osteoclasts, osteocytes, and then just that word osteogenic cell, that's just a term for another term for the stem cell. So let's look at the osteoblasts, and if you just try to draw a representative cell, and just kind of, I think it kind of has that little bump shape to it with a nucleus in it. Okay, so they are going to be producing, or the production of bone in increments or layers is called apposition, so that verb oppose applies here too. So this is just the cells that produce, form, or oppose the bone. Okay, and like I've already said, they're located at the surface of the bone. In the case of alveolar bone proper, they're located in the PDL. So this is just showing you a diagrammatic representation how they're not, this is the bony tissue, this is the osteoblast at the surface. Okay, osteoclasts are much different than osteoblasts. They're huge cells. They have this kind of ruffle border, and they have more than one nucleus, so they're said to be multinucleated. So this would be a diagrammatic representation of an osteoclast. Okay, so as already mentioned, they destroy or resorb the bone. Recall, resorption is destruction of a mineralized tissue. And we're going to see, just like osteoblasts, they're outside of the tissue, and they're going to like create a ditched out area at the surface of the bone that they're resorbing, as shown with my drawing. Okay, and then this is just an illustration showing that they're not in the bone itself. They're in this little ditch at the surface. So with alveolar bone proper, osteoclasts are found in the PDL space. Okay, let's look more closely at the osteocytes. These cells, if you were to draw, they have a cell body with a nucleus, and they have many, many cell processes radiating around the cell body. So that's what an osteocyte looks like. So these are actually in the bone, as it says here, and we'll talk about their inert. I'm not saying they're inactive. I'm just saying they don't move. They're like stationary within the bone, because how did they get there in the first place? That when the bone was forming, some of the osteoblasts, just think that they're slower, they get trapped inside the bone as it's forming. Whereas the other osteoblasts will keep forming bone out at the surface. So these were originally osteoblasts that become entrapped during the formation of the bone. Okay, some additional information I just want you to write about the osteocytes on the side. I know there's not actually room in the chart there, but like I just want to you to understand that these are incredibly important cells in the regulation of bone metabolism. The whole balance of minerals in your bone is regulated by these cells, or what we call mineral homeostasis. So what they do is they kind of send messages to each other, and they also extend send messages to uh, osteoblasts and osteoclasts. So this is just kind of a diagrammatic representation of those cells, and they send out specific messages that stimulate osteoblasts, and at other times they send out messages to osteoclasts. So they're not just, they used to be called like retired osteoblasts that were just trapped in the tissue, but they really have an important function in the homeostatic balance of bone. You know, I mentioned to you that last week that bone is a dynamic tissue. It's constantly remodeling, and we're going to look at that more closely. But these are kind of like the controllers. Just think they're like the operators, like telephone operators, or uh, the center of a computer, what regulates all the other activities. Okay to go on?
Yep.
Okay, so when we actually look at bone, here's a decalcified section. This is what you've been looking at all semester long, where the tooth in situ slide, where it's a decalcified section, meaning the mineral component has been taken out, and then colorful stains are applied to the non-mineralized portions of the tissue. So right here we can see a magnification of a bone trabeculi, and within the bone itself, this is an osteocyte. You can see it inside, and then out at the surface of the bone, it just shows you how an osteoblast, a much smaller cell with a single nucleus, and then a huge osteoclast with multiple nuclei. And you can actually see the ditched out area that it creates at the surface of the bone. Okay, so those are the cells associated with bone. I want to just make sure a couple terms are clear to you in terms of the extracellular matrix. In connective tissue proper, or in the periodontal ligament, there were no mineralized or calcified components. But now in bone and cementum, we're going to see there are non-mineralized and mineralized components, or some terms that are used are organic and inorganic components. So let's look at that in the chart. The organic components are the non-mineralized or non-calcified components. You just have to have that in your mind so that you don't get the two different components mixed up. If I ask you questions on the homework about what are the organic components versus what are the inorganic components. Inorganic refers to the mineralized or the calcified. So I can use non-mineralized, non-calcified for organic, or I can plug in mineralized or calcified for inorganic. Okay. So let's look at this organic matrix. First of all, the name of the organic matrix in bone is called osteoid. So if you just write that in the chart, and this organic matrix is again produced by the osteoblasts. We've already established that the osteoblasts produce the osseous tissue or the bony tissue. Okay, what is osteoid or the organic matrix made up of? It's the same things that you already know about in connective tissue proper. Type 1 collagen, remember that's that strongest non-stretchable fibrous protein. And you know, bone is very important in providing mechanical support. Our whole skeletal system, you know, provides that mechanical support. So it makes sense that the strongest fiber type makes up the bone. And then in addition, there's ground substance that's just the same thing as we saw in connective tissue proper and in the PDL, protein carbohydrate complexes. Okay, so under fibrous matrix, if you just write in type 1 collagen only, and under ground substance, I guess, you know, that it's, remember, this is the amorphous gelatinous part that's made up of protein carbohydrate complexes.
How are we supposed to write this in on this chart? I'm just like a little confused.
I mentioned earlier that I suggested earlier that you have like some little side notes and we just kind of put the main points in the chart. So I'm just going through this one thing at a time, that this is the organic matrix. There's a blank there that says number 1, what is the name of it? And the name of the organic matrix in bone is called osteoid. So in that blank, you would write osteoid. And then under, so what is it made up of? Osteoid is made up of type 1 collagen only under the fibrous matrix portion. And then I guess with the ground substance, there isn't really anything in addition to write. You know, it's nothing different from what you learned about before. We didn't go into all the details of the chemical composition of ground substance other than to say it's made up of protein carbohydrate complexes. And then yeah, I didn't mean to have all this slide pop up at once, so that may be what's a little confusing. So sorry about that. So what does this organic matrix function as? So just on the side, this provides mechanical support to the bone, and it's going to be a framework for the mineralization process to occur. And we're going to talk about how the osteoblasts produce the bone momentarily, but I just want to make sure you understand all the components of bone, and then we'll talk about how it's put together or built. Now on the cementum side, there's also an organic matrix, and it has the same components. So over in that column, I'm going to have you go ahead and write in that it's type 1 collagen and ground substance. In contrast, though, the organic matrix is called cementoid, not osteoid, or sometimes you may see the term pre-cementum. And this is produced by the cementoblasts. So the organic matrix of both bone and cementum is made up of type 1 collagen and ground substance. And in bone, it's called osteoid, and in cementum, it's called cementoid. Notice it makes up approximately 50% of the tissue. Okay, the inorganic components that it's hydroxyapatite crystals. And I'm just, that's all you have to write in the blank is the HA crystals. I'm just going into a little bit more explanation about about it. And hydroxyapatite crystals represents a type of crystalline structure of minerals, calcium and phosphate salts. So it's like this is actually the the crystal organization is, which of course you don't have to be able to plot that out. This isn't a organic chemistry or chemistry class. The formula for calcium hydroxyapatite is this. I'm not going to require that you know it. It may show up on your boards, but most likely not. If you just understand that it's these mineral crystals that are called hydroxyapatite crystals. So this mineral component, or this calcified component, are these hydroxyapatite crystals, and they're identical in bone and cementum. So, and this is roughly 50%, it's maybe slightly less, and that may surprise you that, bone is only 50% mineralized approximately, and so is cementum. So they, what is the clinical significance? They both have roughly the same degree of hardness. And you already know two other dental tissues, dentin and enamel, are more mineralized, so they're actually harder than bone and cementum. Okay. So we have an organic matrix in both bone and cementum. In bone, it's called osteoid, and in cementum, it's called cementoid. And mineral crystals actually form in this organic matrix, which results in a mineralized tissue, bone and cementum, respectively. If you just take that ending, that suffix oid, it means like. So if you take the word human and put oid on it, humanoid is human-like. Well, osteoid is bone-like, and cementoid is cementum-like. It's just what they're called before the mineral component is added to the tissue. Okay, and again, to reiterate this, the hydroxyapatite crystals, just like I said in the introduction to connective tissues unit, it provides mechanical support by making the tissue hard. And both bone and cementum have approximately the same degree of hardness because they're both approximately 50% mineralized. Okay. And I've already mentioned that alveolar bone is vascular. I had shown in this picture under the alveolar bone unit that how some of the blood vessels travel through the bone and go up and supply the gingiva, and some of them enter through the alveolar bone proper and supply the PDL. I've talked about how on the surface on the buccal, and on the lingual, covering bone is a fibrous connective tissue that's loaded with nerves. When your mirror rim hits that, it's very uncomfortable because you're stimulating pain nerves associated with the alveolar bone. Okay, this is a slide we looked at before too, and it of alveolar bone proper, and this shows us the blood supply within parts of the alveolar bone. Cementum, on the other hand, if you look at it right here, there are no areas like this that have blood vessels. There's no blood supply, and cementum relies totally on the PDL for its nourishment. cementum has no nerves, so cementum itself technically is not sensitive. And we'll be talking about that, the clinical relevance later. Okay. So like I said, I'll focus on that later. Okay, the other thing at the bottom of the chart, it says there that both under the PDL, we learned that the terminal ends of the principal fibers that attach to alveolar bone proper and then cementum on the other side are called Sharpie's fibers. So in addition to the components already listed in the chart, alveolar bone proper and cementum also contain Sharpie's fibers. So at the bottom of the chart there, just add that in the blanks, contain Sharpie's fibers. And remember, they're produced by the periodontal ligament fibroblasts. They're not produced by osteoblasts or cementoblasts. And again, they are also classified as type 1 collagen. So we've got two sources of collagen, type 1. The PDL fibroblasts produce the Sharpie's fibers, and then the osteoblasts and the cementoblasts produce the rest of the type 1 collagen that's in the tissue. Okay, now that we've talked about the composition of alveolar bone and cementum, let's talk a little bit about the homeostatic balance in bone. So where it says dynamics of bone, I'd just like you to write this information in there that, bone is constantly changing. In other words, there's physiologic remodeling of bone that takes place. And we're going to see the combined effects of apposition and resorption listed in the two columns there. So when we see a bone at any given time, what we're seeing is the combined effect of apposition and resorption. And in head and neck anatomy, when you're going to be studying your skulls, they're going to stay the same because it's not in a living person, but bone is constantly remodeling. And in disease, we're going to see a net bone loss. Okay, so let's look at apposition first. So remember, apposition, it means the formation of a tissue in increments or layers. And both bone and cementum form in increments and layers. Or we can say they form layer by layer. The term for bone formation specifically is called osteogenesis. If you translate that, genesis means the beginning of or the formation of, and then the prefix osteo means bone. So osteogenesis is bone formation. The general way that it forms is through apposition. It doesn't form all at once. It forms layer by layer. But even in health, there is resorption going on for this overall remodeling process. So you have to think of maybe like a house, and apposition would be building on an addition, and then resorption would be just tearing it down. And then a lot of times there's reapposition. It builds it back up in the same spot. So just think of putting on an addition to a house, tearing it down, and then rebuilding it. Okay, so resorption is destruction of a mineralized tissue. Okay, let's look at apposition first. So I mentioned that bone is formed layer by layer. It's an appositional process. This layer by layer formation is a two-step process. The first step is formation of osteoid. That's where the osteoblasts produce type 1 collagen and ground substance. So over here, if I just take here's an osteoblast at the surface. So when they lay, the first layer that they lay down is just going to be this type 1 collagen and ground substance. Okay, the second step is, I'm just going to show this. As they're laying down a new layer of osteoid, so let's just say they rest at night and the next day they lay down a new layer of osteoid. Notice how they're still staying outside the tissue. The previous one is going to mineralize. So this original layer that was osteoid is now going to mineralize. And that's just mineralization of the osteoid layer. Once you have mineralization, then you actually have bone. So this is a layer of, where the arrow is now, that's a layer of bone. And the layer next to the osteoblast, that last layer laid down, is osteoid. There's always going to be a layer of osteoid next to the osteoblast. That last layer is going to be non-mineralized. Okay, so you just have to picture it's a two-step process, layer by layer. First osteoid forms, and then that collagen and ground substance serves as a scaffold for minerals to mineral crystals, the hydroxyapatite crystals, to form. And those minerals come from the osteoblasts. They pump out calcium and phosphate ions, and they form that hydroxyapatite crystalline structure. Okay, if we go to the top of the next page and compare it with cementum, cementum occurs in the same two-step process. But let's just write in the differences. First of all, cementum formation is called cementogenesis. Okay, and it says there in the left-hand column there that it occurs in the same two-step process. Okay, that same two-step appositional process. But instead of osteoblasts forming osteoid, we have cementoblasts first form cementoid, and then they add the mineral crystals. Okay, so it's all and the mineral component is the same, the hydroxyapatite crystals. Okay, so all you have to do is, it's the same process in cementum, you just plug in different words. It's cementogenesis, the process. The cementoblasts first produce the cementoid, and then they add the hydroxyapatite crystals, which I'm sorry is missing from this slide. And they're this, the mineralized component is just the same, the hydroxyapatite crystals.
I have a quick question.
Sure.
For the first chart with number 2, the inorganic component, is it the same for the cementum?
Yes, it's the hydroxy apatite crystals.
Okay, thank
Yeah.
you.
Sure. So that's what I was just saying here. You have organic matrix formed by the cementoblasts. It's the same thing as the organic matrix of bone, only it's called cementoid. And then, sorry, it was missing from the slide where it says in there that the mineral component is the same thing, the HA crystals. So the cementoblasts would pump out calcium and phosphate ions that would crystallize in the organic matrix, and then you have cement. You no longer have cementum, you have cementum. You no longer have cementoid, you have cementum when the crystals are added. Okay, let's go back to the bottom of the page we were just on and talk about bone resorption. Resorption takes place in a two-step process. The osteoclasts first dissolve the mineral component, the hydroxyapatite crystals, as it says on your outline, and then after that, they produce digestive enzymes that break down type 1 collagen fibers and ground substance. So it's kind of going to be the opposite. First, the mineral is dissolved out, and then the organic is dissolved out. Okay, and all that's the two-step resorption process for bone. And then if we go to the top of the second page, we're going to see for cementum, it occurs in the same two-step process, but I just want you to write in the blank there that it's more resistant to resorption. So I've already mentioned in health, bone is a dynamic tissue, constantly remodeling. We have the net effect of apposition and resorption, whereas cementum is less likely to resorb in a healthy situation. When it does resorb, remember, it's the cementoblack class. And then what stimulates these cells? Again, as I've previously mentioned, it's the eruption of succedaneous teeth. It triggers cementoclasts to destroy the root of the primary tooth during the exfoliation process. And then if there's a pathological condition, such as excessive occlusal forces on a periodontium, can trigger cementoclasts as well. Okay, so in both bone and cementum, there are similar appositional processes and similar resorption processes, but in healthy cementum, the resorption process is less likely to occur. Okay, let's look specifically at the microscopic features of alveolar bone. And again, at the same time, we're going to compare cementum with them. Go ahead and let's go back to the osteocytes. Remember, I mentioned it has the cell body, and then it has numerous cell processes radiating around that cell body. And osteocytes, remember, they become entrapped as bone is forming. They're former osteoblasts. And what happens is the tissue all around them mineralizes, but the space where their cell body is found is called an osteocytic lacuna. Okay, so their cell bodies occupy spaces called osteocytic lacuna. When you translate that word, it means like a little lake. So just think it's a little space where the cell body is found. So just think they just have the So all around them, everything else is mineralized, but they're just trapped inside that lacuna. So just as an analogy, if you're sitting, just picture that if you're an osteocyte and your chair or whatever, wherever you're sitting, that would be your lacuna. If you move out of it, you have that little space there. That's analogous to what we would see in bone. The osteocyte occupies a space called the lacuna. Now, in addition, if the cell processes are also not mineralized, so channels are created around the lacuna, and those channels occupied by the cell processes are called canaliculi. So here would be an example of an osteocyte, and where that space it's occupying, the white space that it's in, is the lacuna, and then where the cell process is that's not mineralized, those are all the canaliculi. Here's an actual osteocyte under an electron microscope. You can see its cell body, and you can see the numerous cell processes radiating around the cell body. Okay, now their appearance is going to vary depending upon the slide preparation. On your outline, it says a ground section versus a decalcified section. So we're going to, I'm going to discuss and illustrate how an osteocyte in its lacuna looks different depending upon the slide preparation. So, and cementocytes, just while we're on the process, how does cementum compare? Okay, the cell bodies of cementocytes occupy spaces called cementocytic lacunae. And they too have cell processes that fill channels called the canaliculi. Okay, but we're going to see momentarily that the number of the cell processes and canaliculi is going to be different. We're going to see fewer. If you actually look at this cementocyte, you can see there aren't as many cell processes, so there won't be as many canaliculi, and their orientation is different. If you notice, they're mainly on one side here, and we're going to, I'll draw that momentarily. Okay, so if you go down to where it says decalcified section, okay, a decalcified section, as I mentioned, is where the tissue that's...
Myers,
Yeah?
can you go back to that last slide real quick?
Sure.
Okay, thank you.
Welcome. Okay, so a decalcified section, you know, what will help you recognize that is that it usually has bright colors like purples and pinks, or deep reds depending upon the stain. But before, what's happened here is all the minerals been dissolved out of this bone, and then just the organic materials are stained. So in here, the bone has a pink color to it. In contrast, a ground section, the histotech who prepares these slides, actually they have a machine that takes hard tissues, For example, they can take bone. They can also take a tooth, and they have a machine that just grinds it into thin sections. Just think of like a little almond sliver, and they put it on a slide. So you're actually looking at the mineral part of the tissue. And so this is what bone looks like. All the white areas are where it's mineralized, and all the dark areas is where it's non-mineralized, or where there are spaces, or in other words, those lacunae. So ground just is the past tense for grind. They ground the tissue to prepare the microscopic slide. So this still has mineral. The decalcified tells you it does not. Okay, so what do we see with the decalcified section for both bone and cementum? With Halloween coming up, I use the analogy that an osteocyte in its lacuna looks like an eyeball. So if you just draw a circle to represent the lacuna, and then the cell nucleus of the osteocyte would be analogous to the pupil. That's what an osteocyte looks like in its lacuna. And it's shown in the smaller scale on your outline, but there's the lacuna, and then there's the nucleus of the osteocyte. So that's how on a ground section the osteocytes appear. In contrast, with a ground section, you don't see the osteocyte. You don't see the cell body. You don't see the cell processes. Instead, you see the lacuna with the canaliculi. So again, with the Halloween analogy, on a ground section, the space that was occupied by the osteocyte, the canaliculi that were occupied by the cell processes, are not mineralized. So they show up like a dark space. So here's the osteocytic lacuna is going to be just a dark space where the osteocyte was. And then the canaliculi are just like the little legs of a spider. And with an osteocyte, they're going to radiate completely around the lacuna. So it looks like a spider. An analogy for this would be just like when you put a footprint in the sand, when you're at the beach and you put a footprint in the sand, your foot is not there, just the space where your foot made the imprint is. And just think your toes would be kind of like the cell processes or the canaliculi. Or another analogy would be with a fossil. With dinosaur fossils, we're not actually seeing the dinosaur, we're just seeing an imprint in rock or mineralized tissue where the dinosaur was located. It's the same with an osteocytic lacuna. So here, if you look in this lower left picture, this is a ground section, and all of these black areas, like right here, this represents an osteocytic lacuna, and the black extensions are the canaliculi. We're going to record in 3 characteristics of the osteocytic lacunae and their canaliculi in bone. So how do I know that this is actually bone from this ground section? First of all, the lacunae are dense. In other words, they're more numerous. There are a lot of them. They're all over in this tissue. And in this particular slide, it may not be as apparent, so I'm going to go back to a lower magnification here. But notice how these are all osteocytic lacunae, that they're neatly arranged in a row. See how they kind of go around in a neat circular pattern with this concentric lamellae. And this is just a magnification here. So they also have a neat arrangement. They line up in rows. Here you can see it a little better at the top here. And then the most distinguishing feature is how I've shown it with the osteocytic lacuna I already drew, and we can see right with this one that I've encircled that the canaliculi radiate uniformly around the lacunae. So those osteocytes seem to communicate in all directions, because remember their cell processes are in all directions in these canaliculi. So I can more easily tell bone on a ground section compared to cementum, because we're going to see the lacunae and canaliculi look different. Okay, so let's do the comparison with cementum now. This picture right here, this shows you cementum. It's at low magnification, but we can see we can see the osteocytes in their lacunae in the bone, and over here on the cementum side down in this area, I can see little cementocytes in their lacunae. And I want to point out that if I were to like zoom in really closely, on a decalcified section, a cementocyte looks just like a osteocyte. It looks like the little eyeball. You can see so it looks just like the little picture that you have in the outline. So when I'm really zoomed in, I really can't tell. If I were just zoomed in on this, I wouldn't be able to say whether that's a osteocyte in its lacuna or a cementocyte in its lacuna. So the appearance is the same. Here's the osteocyte in its lacuna again. I couldn't really tell them apart. But when I take a ground section of cementum, I can see a difference in the cementocytic lacunae and their canaliculi. So this is a representative of cellular cementum that has cementocytic lacunae. And you should notice in the picture that there aren't as many of them. So there are fewer cementocytes and thus fewer cementocytic lacunae in cementum. In addition, they tend to be more scattered. These aren't really lined up. Here's one here, one here. They're not lined up in rows as neatly. And then finally, if you notice, the canaliculi are oriented to one side. So here I'm going to draw a cementocytic lacuna, and its canaliculi are oriented to one side, just like when I showed you that electron micrograph of the cementocyte, the cell processes were oriented to one side. So they don't go uniformly around the lacuna. They're on one side. And you can generally tell which side is the PDL side. I mean, this is a ground tissue here with this is just the cementum, but the PDL side is on the right because that's the source of its nourishment. So they're always going to reach out to the side of their nourishment. So again, I know this is a ground section, so I wouldn't see it just like this, but the PDL would be on this side because this is where these cells get their nourishment. Cementum's avascular. So for example, if I had you in class and I was passing out candy, you would, and I said you had to come and reach for it, you would reach out to the side that I had the candy on. So that's just how the cementocytes are in their lacunae. Okay, so we've talked about where the osteocytes and the cementocytes are in their lacunae and how they vary in their appearance depending upon the type of slide preparation. All right, let's go to the next page. And we're going to look at structures that are specifically associated with apposition, the layer by layer formation of the bone in the two-step process. Okay. Last week I already told you that there are lamellae in bone, and then we're going to add another structure. There are what are called arrest lines or resting lines. Okay, so lamella is a Latin term which means a thin plate, and for our purposes it actually just means to the individual layers in the bone. So bone forms layer by layer. And we're going to review, remember, in compact bone, we can see both longitudinal lamellae and concentric lamellae. Other structures associated with apposition are referred to as the resting lines, or they're on your handout it says they're called arrest lines, incremental lines, appositional lines. They all mean the same thing. And what are they? They're actually lines that separate the layers. And what they represent are periods of rest between periods of activity by the osteoblasts. So those osteoblasts are builders. They build so much during a day, and then let's just say they rest at night. As a general rule, unless you're someone who works third shift, we're generally diurnal creatures. Just writing that word in. Diurnal means that most of our activity is during the day, whereas there are other species that are nocturnal, such as bats. So our cells tend to follow a diurnal appositional activity, where they're active during the day, building the layers of bone in the two-step process that I mentioned, and then they rest at night. So if we look at this illustration, let's label what this is showing here is this is a developing tooth root right here. So that's a developing tooth root. We're way down, and in this area right in here is a developing PDL. Notice we don't really see the principal fibers yet, but we just see different cells and blood vessels in the PDL. For example, here's so we don't see those clear-cut interstitial areas and areas of principal fibers yet. Okay, pictured on the far right is alveolar bone proper. Okay, remember the alveolar bone proper is the part of the alveolar bone that forms the tooth socket. So here we have a developing tooth root and it's developing bony socket. What's not shown, which would be continue to the right, would be the spongy supporting alveolar bone and the compact alveolar bone. Okay, what you're seeing here that's shaded in, I'm just going to put X's in them at the top, are 4 layers of bone or lamellae. Okay, those are, and if you notice, they're oriented like this, like pieces of plywood. They represent the longitudinal lamellae. So they are longitudinal lamellae, and they're shaded in gray to show that they are mineralized. Okay, right next to the PDL, alveolar bone proper is going to be made up of longitudinal lamellae, and that's because those layers are getting their blood supply from the PDL, where I circled those 3 blood vessels. I'm going to have you draw in alveolar bone proper is also consists of concentric lamellae. So where the white space is, I'm just going to draw in a couple of them. I'm going to draw a central blood vessel. And then I'm going to go ahead and draw the concentric lamellae. So here. Remember, this alveolar bone proper is dense, solid bone. So lengthwise are the longitudinal lamellae, and in deeper are the concentric lamellae, where the bone has a blood supply deeper in the tissue. So notice separating these layers are lines. If again, I'm not going to go all the way down, but here is a kind of faded out at the bottom there, but that is one of those resting lines, or these borders of these circular layers represents a resting line. So what happened is that this layer of bone formed, then where you see this line, that's where the osteoblasts rested, and then the next day they formed this layer. So we're always going to see resting lines separating the lamellae, whether they're longitudinal or whether they're concentric. Okay, the other thing I want to illustrate here is that the last layer laid down, I'm going to color it in orange, and it's not shaded on your drawing. Okay, that is osteoid. The last layer that the osteoblasts form, remember, is the organic matrix, just type 1 collagen and ground substance. The very last layer has not mineralized, so that's not bone, that's osteoid shown in orange. This arrow right here is pointing to it. And then notice on top, of the osteoid at the surface, not in the bone, and that arrow right here is pointing to 1. Those are all the osteoblasts at the surface. Okay, so we're going to see layers of bone separated by resting lines or arrest lines. And at the surface, we're going to see the osteoblast, but always the last layer is not actual bone, it's osteoid. So those are all osteoblasts lined up in the PDL that we've already discussed as being cells of the PDL. Now that's the arrangement for alveolar bone proper.